All right, we are back. Today we're going to talk about how do muscles contract? How do they contractumulate? Um, and we went over. Oops, let me get rid of this. Went over the basic anatomy of a muscle, broke it down into all of these and all these different stages, and I just want to kind of go over this again real quick. This little part. So we've got our muscle cell with our myofibril. And we have these things called sarcomeres, which actually look something like this. We've shown these. So this little thing that I've drawn here, this little sarcomere, relaxed versus contracted. It's made out of actin and myosin. That's what makes up your skeletal muscles. So this is a sarcomere. We got this Z line here. I don't care, these other parts. The A band, maybe know that one. That's actin and myosin. It's pretty easy to remember the A band there. Yeah, and this is what they actually look like under an electron micrograph, under a microscope image. And you can see just how organized and repeating they are, and that's why we see these striations. So where you see the Z line, that's actually lighter in that area, and then it's darker and more dense because all the fibers of actin and myosin in the center. I just wanted to show that little picture there. Okay, so I went over that. How does this actually work now? That's the anatomy of it. What's the physiology of it? So this is the actin filament. I've drawn it in one of the colors that myosin was drawn in. So I've drawn myosin in different colors. So this is one of the actin filaments, the one on the top, let's say. So that'd be like this one right there, and then we're looking at these myosins underneath it. And so this is the myosin right here with some of his other buddies. There'd be way more of them. There's something like 300 myosins in each one of these bundles. So don't worry too much about that being totally accurate. Okay, so now how does this work? Well, the general idea is that this thing, let me grab this. These little heads are going to move like this, just like that. And when they do that, they're going to grab and like move that down and move that down. So it's contracting, it's moving this actin filament, sliding the actin filament along it. That's the general idea. How does this actually do that now? All right, let's first, we're going to have our good old friend ATP come into the play. And ATP, we're going to draw it as an A, and then we're going to draw it as 3 little circles. And phosphate, we'll draw it as greens. Boom, boom, boom. Okay, so that's ATP. It's ATP. Just label that like so. All right. Well, ATP binds to this. And when it does that, the myosin head now releases from the actin filament. So we'll keep it, actually we'll keep this one just the way it was. Sorry, let me get rid of that. And that, that, and that. And I'm going to copy all this. Yeah. I'm just going to copy everything and get rid of what I don't want. Okay. So ATP binds. That's what we're talking about right here. ATP binds. All right. ATP binds, and when it does that, what's going to happen is this myosin head is going to release from the actin filament. It's pretty interesting. Look, why are you doing me like that? Why are you doing me like that? And so now this myosin head has been removed. It's now disconnected from the actin filament. So when ATP binds, when ATP binds to myosin motors, it releases the myosin head from the actin filament. Pretty interesting. So the binding of that energy, of that ATP, releases the myosin head from the actin filament. It's pretty interesting. Okay, now what happens is this. I recopy all that again, and you could just draw that actin filament as like just a line or a however you want to draw it. It doesn't have to be this detail, but actin looks like a little twisty helix like that. So then what happens is this ATP hydrolyzes. It removes that phosphate. It's still kind of attached to the myosin head, but it's now not a part of that molecule. And what this causes is rebinding of the myosin motor. So the myosin motor will now rebind, let's say like so. Okay, so it's called how do we cut something? Hydrolysis. So ATP hydrolysis into ADP causes myosin to rebind the actin filament. All right. And then last but not least, we get something, one of my favorite science terms. So this is ATP to ADP hydrolysis, hydrolysis. That's that step. ATP binds, ADP to ADP hydrolysis, and then here we have what is known as the power stroke. So I'm going to redraw this little myosin head. So what happens is this guy, this ADP, I can't remove it all for some reason. This guy leaves, and I'm just going to get rid of that. And this guy leaves. And what happens is this guy kind of bends back like that. You see how that one's kind of straight, it now bends back. So the power stroke has occurred. So release, let me draw it like this, actually. So release of ADP and phosphate causes myosin head to bend back. This is called the power stroke. Hit him with that power stroke. It's interesting because it's not the hydrolysis. See if I can, this is a little, oh boy, what did I just do? You know, it's not this hydrolysis, which is like actually the where we would get the energy. This whole system is kind of counterintuitive to me. I want that on the top. There we go. Like it doesn't really seem to make, this isn't the way I would do it if I was designing the universe. This seems counterintuitive. So you're telling me when we bind the energy, that releases the myosin head, and then it hydrolyzes it and rebinds, and then the energy molecule falls off, and then it does the power stroke? That doesn't really seem to make sense. I would have it where, like, the other way. Like, once it falls off like this, that's when the head should come off. And then when it binds ATP, that's when it should attach. And then when it cleaves ATP, that's when it should do the power stroke. But that's not how it works. So it's kind of counterintuitive. This whole thing is kind of counterintuitive. And it leads to something really interesting that we're going to talk about. So this part, right, it's really important. To release myosin from actin, it requires ATP. So when you die, eventually you run out of ATP. Okay. Eventually you run out of ATP when you die. So your muscles will be locked in this state. They will be locked into one of these, probably this state. They'll be bound and they'll have lost ADP and PI. They'll be locked in one of these two states. When you die, after a few hours, your myosin will be attached, will be bound to actin filaments. Okay. And this will, so after a few hours of being dead, you're going to have all your myosin motors bound to your actin filaments. And there's a special name for that. Rigor mortis. You are locked into place. I don't know if you've ever found a corpse of a human, maybe an animal, and that boy would be like, it's pretty interesting. And this is the reason for it, is we've run out of ATP because the cell's not alive to make any ATP. And so now all of our myosin motors are either locked in this state or this state. Eventually what'll happen is decay. We'll have decay of these filaments and these motors. Eventually, decay will break down the actin and myosin, from bacteria and things like that. And the corpse will go limp again. We're going to have 2 activities this week, actually. Yeah, no real discussion. No. We're going to have 2 activities, 2 discussions. We'll see how you guys do. Because I got 2 cool activities this week. I want to talk about the amount of ATP that's used by your bicep. And then we're going to do this one as well. This rigor mortis activity. So get ready for that. It's quite a bit. But it's all pretty interesting. And then you can, if you ever find a corpse, you can determine if the relative time of death. You're going to learn how to be a little bit of a detective, which is pretty interesting. All right, so that's one really important aspect of cellular physiology. Now, let's see. How do I want to do this? Okay. Let's draw a little muscle cell. So there's another important aspect I want to talk about, because it's interesting, because this is one of the main reasons why, another reason why we need calcium. And we're gonna look at this actually in the lab too a little bit. So it's pretty cool. All right, so here we've got a muscle cell, and a muscle cell has what? Multiple nuclei. So maybe let's draw in a couple nuclei. Like this. And then we've got some muscle myofibrils. We're just going to draw 2. And yeah, I'll draw 2 like that. And these things are full of what? Sarcomeres. 2 myofibrils. Okay. So this is muscle cell, and these are myofibrils. Myofibrils full of sarcomeres, which are actin and myosin, right? I'm not going to draw them in, but these are our striations. These are our contractile doodads. These are these, which are full of this, right? Which requires ATP to disconnect. And let's actually put this text down here, just so we can have this be clear. Myofibrils full of sarcomeres. Okay. Interesting thing about this is we kind of got a little membrane channel that comes down on muscle cells. Let's say a couple of them go like this, and they go around the myofibrils. Let's do one there, and we'll do one here. Okay. So this is like an interior muscle cell membrane. It's called a T-tubule, but I don't care that you know that. This is like membrane that wraps around myofibrils. This is important for carrying signal, and I'll show you why. Okay, so we got some signal being able to be carried down these channels. Well, where's that signal coming from? Well, it's coming from a neuron. We've not talked too heavily about neurons, but this is a neuron coming down at a neuromuscular junction. And we'll talk about how neurons work and how they make synapses. So this is the axon of a neuron. How do you fire your... How do I do this? Move my muscles. How does my brain tell my muscles to move? Or my spinal cord to tell my muscles to move? Well, that neuron sends a signal. Down like this. And then it goes down in either direction down these T-tubules. It's an electrical signal. We'll talk about how that works. Okay? But how does this result in muscle contraction? That's the question. All right, well, we actually have a very specialized type of endoplasmic reticulum that surrounds these. And I'm going to draw it. It kind of weaves and wraps around these, kind of like a spider web. I'm not sure exactly how I want to draw this because it's kind of crazy. Yeah, maybe I'll just draw like that. I'll just draw like wrapping around like that. That works. So this would cover the whole myofibril, but something like that. And this is specialized endoplasmic reticulum. Okay, that's what that is. Specialized endoplasmic reticulum. It's called the sarcoplasm. And it wraps around each one of those myofibrils. And I'll pull up a picture of this in a second so you can see it. See a little better drawing of than the one I have. Okay, that's what this is. And what this electrical impulse does, this electrical impulse stimulates this sarcoplasmic reticulum, and it tells it to release, what color is calcium? Maybe this color. Tells it to release calcium. So now we have calcium being released. Okay. So the impulse, it's called an action potential from the neuron, stimulates calcium ions, Ca++, calcium ions, Ca++, from calcium ion release from the sarcoplasm. All right. So this would be like, yeah, signal is transmitted. Just trying to kind of put an order to this because I went slightly out of order. So this would be like 1, 2, and then 3. It's transmitted down those membranes, and then 4. Okay, 1, 2, 3, 4. Get your woman on the floor. Gotta get up to get down. You know what I'm saying? That's how we do it. This is right here is how we get down. Okay, well, what the hell does that? Okay, so we get a nerve impulse, and that releases calcium from some weird, goofy ER that's wrapped around it like a worm. That makes no sense. All right, let me pull up this picture so you can get a little idea of what this looked like. A little better version. This is what I drew it from. This is a reference I used. You can see how much better it is than mine. But this thing has a lot more details on it than it just makes it a little too busy. But this is what I'm talking about with these ER, these sarcoplasmic. They look like spider webs. And you can see we got this nerve impulse getting sent down these T tubules, which stimulates calcium release from the ER, the sarcoplasm. Another interesting thing you can appreciate is that these sarcomeres and these myofibrils, there's a bunch of them. Like each one of these that you see here, these are your thick and your thin filaments. You can see that they're like three-dimensional. It's pretty nifty. Okay. Well, why do we need that calcium? Let's go to this boy. I'm gonna copy this boy right here and bring it down. Okay. Why do we need calcium? All right, I'm gonna add another layer to this. I'm gonna get rid of this little ATP. We don't need that. And I'm gonna get rid of that and that. And I'm just gonna clean this up. I just don't want to look at all that, but it's all there. Okay. All right, well, actin filaments normally, they're not free like this. So they're actually encased in another little protein that wraps around them like this. I hit my T with my mouse, and so I couldn't draw anymore. And then this filament also, you see it kind of where I messed up on this filament, didn't draw it exactly correctly. That doesn't look that great. That's okay though. So these, it's actually made up of a couple different proteins. But what these do is they lock the actin filament up. They block it from myosin being able to bind. Like so. And I'll name these here in a second. Okay? Something like so. Okay, so this is this kind of long, thin one. I figure out the best place to label it. This is tropomyosin. Tropomyosin, and that's kind of a great name. And this is troponin. Okay? What do these got to do with calcium? Well, it turns out that troponin, let's go copy that, put that down there, save that for later. So now we've got no calcium. This isn't with no calcium. No calcium. No calcium present. All right. Then we have calcium binding. Oops. We got our calcium molecules coming in and they bind to troponin. So now we got calcium, and some of them are going to bind to this troponin. And when they do that, they move these little fibers. They basically slide these little fibers, and this is going to be kind of tough to draw. So what I'm going to do, I'm going to move these all to the back. Yeah, that's pretty smart. Look at you, Tori. You're like a genius. Still taking a while. It's taking a while. Nope, that's not what I wanted. Don't know how to select that tool outright. There we go. Just do the good old click and hold method. So when calcium binds, it causes troponin and tropomyosin to shift their position, which reveals the myosin binding site. And that's why myosin is able to bind. Myosin motors are then able to bind and begin contraction. It's not some, it's a little bit, it's just like saying, it's a little bit of magic, honestly, because it says, all right, I'll just keep it like that because I want to keep moving. Yeah, nah, I'll finish the job. I only got like 5 more to do. It sucks I gotta do these calciums though. Okay, you're bugging me. Why is it doing that? It won't let me get that one, sorry. I know you care. Okay, that one can stay. Okay. All right, so the calcium's now bound, so now we've moved those filaments, and now our myosin head can bind. I know there's a lot of work just so we can have our myosin head bind. So calcium binding, I'll draw it in pink. Calcium binding slides tropomyosin around actin filaments, exposing the myosin binding sites, which I don't really have depicted, but now you can see myosin can bind here. It's unfortunate that that is right there, but you get the idea. Now it can bind to the actin filament. It doesn't bind to tropomyosin. It binds to the actin filament. Okay. Man, sorry, I feel a little out of it, dude. Okay, no calcium present. Let's add some notes there. Myosin binding sites are hidden, are blocked. are blocked by tropomyosin. Okay, so this is one reason why we need a lot of calcium in our body, not just for our bones, but for our muscles to function. Okay, and another reason why we need ATP, or why rigor mortis sets in, is because we need ATP to pump calcium back into the sarcoplasmic reticulum.